Hello and welcome to DNA Today. I'm your host, Kira Deneen. DNA Today informs you on what's happening in the genetic world. We're talking about news stories, lessons, all to keep you updated and educated on what's happening in the world of genetics. Today in the show, I interviewed Denise Anamani, the academic coordinator of laboratory education for students in Diagnostic Genetic Sciences Program here at the University of Connecticut. I also have a few news stories to follow up after the interview. But before I get into either of those, I want to talk briefly about a genetic-related presentation presented last week here at UConn. And this was by Dr. Mike Shapiro from the University of Utah, Department of Biology. And he presented Endless Pigeons, Most Beautiful, Genetic and Developmental Origins of Diversity. And he talked about a lot of different things. And the main idea here was that there's very little known about the genetic causes of phenotypes, and we don't know if what genes specifically cause all phenotypes, or what they are in coding regions. That's the part of the DNA that actually codes for proteins, because they're parts of DNA that doesn't code for proteins. There's not a direct correlation between, the base pairs and proteins, and also regulatory regions of how we are regulating the DNA and using different proteins for that. So he kind of touched upon a lot of these ideas. And his research looks at the differences between pigeons. His research is all focused on pigeons, and he goes all the way down to the genomic level by sequencing genomes and highlighting differences between them, between two different types of pigeons. And he finds the region of the genome that birds with common traits share. So if they have a common trait and there's a part of their genome that is similar or identical, then we can attribute that part of the gene or to that specific phenotype or trait that they exhibit. And so this is what he really got into. A funny, interesting little tidbit he shared, which I didn't know, that in 1859, the editor of Charles Darwin's book, The Origin of Species, the editor feared that his subject matter of the origin of species was too limited, and he suggested that he should focus his book on pigeons, and that it was better for the mainstream. So Dr. Mike Shapiro kind of brought this up on how he's almost taking his advice here. But Darwin studied pigeons a lot, and he had a lot of drawings that are very accurate when compared to pictures today of what pigeons look like. Back then, obviously, people couldn't just Google what a certain pigeon would look like, so they looked at his drawings, and looking at them today, they really are accurate. Another person that's famous for working with pigeons is Thomas Hunt Morgan, who is more famous for his work with Drosophila, or fruit flies. And Dr. Shapiro shared this interesting trait that pigeons exhibit. And this is having feathers on their feet, and this is known as muffs. And the range of the length of these feathers is very large. Some of them are, just little tiny feathers, whereas others are almost longer than the body itself. And Darwin actually commented on this trait, and he said, quote, "So heavily feathered, they almost appear like little wings." He also said, quote, "Pigeons with feathered feet very generally have the base of their outer toes connected by skin." So a lot of things that are not new to research, but he's kind of expanding upon a lot of ideas and finding specific traits and trying to find the gene that encodes those traits and, encodes those proteins, which then makes that trait. And I'll have a link in the show notes. You can find on the website if you're listening live. That's dnapodcast.com. And it will go to Dr. Shapiro's lab, so you can learn more about his work about pigeon genetics. And it was a really interesting presentation. There's quite often presentations here at UConn about genetics, free for students. You can just walk in, take a seat, and get to hear from these really amazing professors and researchers and doctors, and hearing what they're doing and really what is happening in genetics, and hear specifically their research. It's great opportunities. I'll try and tweet them out as I find out about them, but definitely check that out. If you're a UConn student, you can check out the Daily Digest. They're usually listed there. That's how I find out about them. Definitely stay tuned with all that. And now I want to get into my interview with Denise Anamani. I'm here interviewing with Denise Anamani. She's the academic coordinator of laboratory education for the students in Diagnostic Genetic Sciences Program with a molecular diagnostic concentration. She works in the Department of Allied Health Sciences here at University of Connecticut. Welcome to the show, Miss Anamani.
Thank you. I'm happy to be here.
So I want to start out with asking you what Diagnostic Genetic Sciences Program and also the Medical Laboratory Sciences Program, because you're involved in that here at the University of Connecticut, what do these programs offer students?
These programs are professional programs, meaning that students are being prepared to enter their profession immediately after they get their bachelor's degree here at UConn and work in diagnostics. So diagnostics really differs from research in that you are involved in laboratory testing with patient results coming out as the final product. Whereas in research you are hoping to answer questions and put all that information that you gain into the general knowledge about a specific question that's out there in science. So there is a difference there, and so these programs are preparing students to really be able to sit for a national certifying exam. They become a certified technologist, and they work immediately with really excellent jobs. The job market is very good right now for people in the laboratory sciences, whether it's genetics or medical laboratory sciences, where it might be microbiology, hematology, or one of the other sections of the laboratory. As people are getting much older and retiring, and as everybody is living longer, there's a really big need right now for laboratory technologists. So our students are usually hired immediately, and often they all do internships their last semester of their senior year, and they're often hired by the people where they intern. So if they intern at Yale University, they are sometimes hired by Yale in the laboratory. So it's very cool, it's very interesting, and we have 100% placement for the students who want to work immediately, and the others go on to grad school. They sometimes go to medical school, PA school, genetic counseling, you name it, they will go into many different areas, and they're well prepared for that as well.
So the program really does, as you said, help prepare them, and if they want to, they can go right on to the next phase of their career.
Correct, right.
Wow.
It's not, so they can either have it be a sort of a terminal thing where they enter the profession and stay in the profession as a laboratory technologist, move their way up to maybe a supervisor, manager in the laboratory, or they can move on to do other PhD or other doctoral level or master's level work.
And how does UConn's program compare to other schools' program for like BS?
For the Diagnostic Genetic Sciences Program, we were the first one in the nation, and we're the biggest one in the nation.
That's a stat right there.
And there are very few of these programs because they're small. We accept 16 students, that's all, into our program. So it's fairly expensive as far as like the resources the students use, expensive reagents in the laboratory, a lot of almost one-on-one with instructor-student. So it's very sort of expensive, if you will. So some universities won't support these programs, but thankfully UConn has been very supportive of our professional programs.
And you said 16 students are accepted per year into the program.
Correct.
How many people are applying?
We usually get around 40 or so applicants. It is hard for students to find us, and that's one issue is we have a hard time marketing that we even exist, and that most students don't even find about us. Sometimes they don't find about us till they're juniors or seniors, and then it's too late because they will have to extend their time at UConn. So we're hoping freshman and sophomore level students find us and can apply as a sophomore and be accepted as a junior into our program.
Wow. So what are your roles in these programs?
So I do the molecular diagnostic, the lectures, the laboratories, and also a seminar type course. Other instructors do more of the cytogenetics teaching, and together we are just a very small group of instructors that work with the students. I also am the coordinator for laboratory education, so what happens is when students are getting ready to go and do an internship, they visit various labs, and I'm the one that sort of coordinates where they will be placed when they do that internship. And we do have internship sites all over the country, so it's not just down the road in Connecticut, but it can be in Montana, it can be in Louisiana, it can be far away.
So this is kind of like if kids can't get a study abroad opportunity because a lot of sciences you have to stay on campus.
Yeah, that's hard.
It's kind of their opportunity.
Yeah, it's a really excellent chance to get away, and you're working side by side with technologists in a laboratory for 6 full months just doing that. You're not coming home and doing homework and reading, but you're actually working side by side. It's just such a great experience, and when you're done with that, you have that real world diagnostic work experience, if you will, and that's why you're very marketable at the end.
As well as building networking and meeting people building connections, and as you said, a lot
Absolutely.
of kids from the internship end up working there as a full-time job.
Right.
after.
Yeah, during the time that the students are doing their internship, it's kind of like a job interview, if you will. The student is impressing the people there, and they're kind of training the student in the way they do things as far as their systems and how they log specimens into their specimen logs and all that stuff. So by the end of that 6 months, it would be smart for the site to hire you because they've already got all that training under.
They've already invested all the time. They might
Yeah,
as well keep
exactly.
it going. Yeah,
Right.
definitely. So you've also worked as a laboratory technologist for 15 years in clinical molecular pathology, immunology, and chemistry. How much of your experience was directly related to genetics?
So for the 15 years that I was in, I worked at Harvard Hospital in the laboratory. The first half of that was in the special chemistry, they called it, doing immunological assays. And then the second half of that was in the DNA diagnostics laboratory. So about 7 years was full-time in the DNA lab. We started off as nothing but having an empty room, and we were able to fill it up, get some instruments, learn how to extract DNA, manipulate DNA to learn about human disease, learn about mutations. So it was really nice because I was at the very beginning of that whole new discovery. And so then at the end of that time, so during that time I was working on my master's at Central Connecticut State University. And I didn't really think of anything specific I was going to do once I got my grad degree. I was just thinking I wanted for my personal enrichment to go back to school. I had little kids, but thankfully my husband was able to take care of them, so I did my part-time master's. And then right after I was done, this opportunity at UConn opened up, and I interviewed, and they hired me because I had that clinical experience.
And which is so important that we keep talking about that in a career like this, that you have that experience in the lab.
They want you to be somebody who did work in the lab, understands what happens in the clinical lab as far as all the quality assurance and all the stuff. There's a lot to a clinical lab experience that's very different from a research lab experience. And so they really wanted that as someone who could be an instructor here for the students going through this program.
To be able to relate with them and help them
Right,
through the
and bring those
process.
stories, to them of what really does happen out there.
Definitely, yeah. So you've had a number of publications, one involving the expression of the protein HUCP2. Can you explain kind of what your colleagues and you discovered in this research and what your conclusion was?
Yeah, we were looking at these uncoupling proteins in Drosophila, which are fruit flies. And the researcher used fruit flies as her model system. And she was looking at lifespan and how that might be related to disease. And so she was treating the flies to different media, different foods, and they were, some were being fed very high caloric foods, others were being fed very low caloric foods. And she was looking at how that affected these different genes in the flies. Some of them would die very soon if they were fed too much or the wrong type of food, and some would live longer. And so she was studying that kind of mechanism. She was also looking at Parkinson's disease and diabetes. So there were different fly strains that had different genetic makeups, and she was trying to determine how much she could gain as far as information from these models that could be translated to human disease. And so she's still actively working on trying to really find a cause relationship that she can determine in the flies relating to those human diseases.
So this project has been going on for a long time.
Yes,
then.
and it is still ongoing, correct. I was just there for a very short while helping her with the work she was doing, and it's still a lot of ongoing work. There's much research in that area.
Definitely, cool. So how did you become interested in pursuing genetics as a career?
Well, I honestly didn't really even think I was going to pursue genetics as a career, and I was hired, when I was in the Peace Corps for a couple of years. I was away. When I came back, I just needed to work, and I got hired by the Harvard Hospital lab as a part-time person. Didn't really know much about lab work, and so I was totally trained on the job and worked in that, like I say, in that chemistry section for a while. And then when a new director for molecular diagnostics came, I started to work under him, and I learned everything there. I really didn't know much about what genetics was all about until I started working in it. Then I was like, yeah, this is it. This is awesome.
That was kind of your aha moment.
That was my aha moment, exactly.
Yeah, and so you got your master's at Central Connecticut.
Correct.
And that was biology?
Yeah, molecular cell biology.
Okay,
yes.
so was what portion of that was focused on genetics, or it really?
It was all
okay.
genetics. Yep, it was all about genetics. It was about learning more about what happens, the mechanisms for transcription, translation, really getting into the labs were looking at different laboratory techniques and how you determine what's happening with DNA and RNA in various organisms. So it was more of a research-based kind of master's, but it was perfect for me because I had worked in the clinical lab, then did that, so it made me more of a well-rounded person in genetics. So it was perfect.
That experience helped you through the education and everything.
It was great.
Yeah. And we both attended the grand opening of Jackson Laboratories for Genomic Medicine in Farmington, Connecticut. Critical conference. There were so many big names there, and a lot of Nobel Peace Prize winners presenting. Is there a presentation or new research that stands out in your mind from the conference?
Well, I was just struck by how much they talked about the microbiome and how much they talked about our normal gut flora and how when that is messed up, so much disease can happen. I was really astounded. 3 or 4 people touched on that. So that was obviously to me a very big topic right now in genetics is what's going on with our gut flora, what is normal, what is abnormal, and how does that relate to different diseases. I was really, really amazed by that. That was the
I was
one
very
thing
surprised
I was struck
it kept
by.
coming up. Yeah.
It kept coming up. So that's obviously a big area of research and interest right now with these researchers. So that was the thing that really struck me. I hadn't really thought it was that big of a deal, if you will, and now I realize it is.
And how it's also related to genetics and it's kind of expanding from there.
Yep.
Definitely. Any other hot topics that seem to be appearing in genetics and really rapidly growing?
Well, I mean, there's no way you can not think of infectious disease right now in the climate that we're in. So, I mean, knowing more about how different viruses and other organisms infect us, how we can fight them off. I mean, it doesn't seem, when you think of genetics, you think of like diseases like diabetes or something, but the infectious diseases are really important. And in molecular diagnostics, some laboratories are very, very big in looking for infectious organisms and deciding what strain a person might have and being able to treat them correctly by knowing that information. So I think we're going to see more and more of people working in genetics with microbes, whether it's the microbiome, whether it's like these other viruses that we're seeing that are starting to become more of a problem in the whole entire world. That's an area that's growing, and it will grow.
Definitely. Yeah, there was quite a few things mentioned during this presentation for the Jackson Labs, and the location really is beautiful. That it's a brand new site, and they're just rapidly growing. I think the clinical lab just started being active, I think, last week. That they finally set things up.
They're barely even starting it. Yeah, it's brand new. Beautiful.
Yeah. And there's actually a UConn DGS alum that is working in the clinical lab, which is very exciting for UConn to see that students
she has
really
a great
propelling.
position. Excellent.
Yes.
A manager of clinical genomics.
Yes.
Pretty cool.
Talia. Yes.
Talia,
Yes. That was a fantastic day. Just for me personally, just being able to network and meet a lot of people and just be exposed to really what's happening in genetics and everything. So thank you so much for being on the program. We really appreciate hearing what you have to do and how you're involved in genetics, especially here at UConn, and your experience with all that. So thank you so much. Really
You're very
appreciate
welcome.
it.
It's great. Thank you for interviewing me.
Yes, thank you.
And now jumping into the genetic roundup of the week. Our first news story is talking about epigenetics and how it relates to binge drinking. So researchers may have identified epigenetic proteins that contain or that change caused by binge drinking. And this is a discovery that could lead to treatments for alcohol-related liver diseases. The lead author of the study, Dr. Shivendra Shukla from the University of Missouri School of Medicine, shared the effects of drinking. Quote, "We know that chronic alcohol use is damaging to the liver, but binge drinking amplifies that damage." End quote. So there's so many negative health effects that heavy drinking causes, especially binge drinking, as Dr. Shukla pointed out. And one of the most common causes of liver failure is excessive alcohol consumption. And once a person causes long-term liver damage, it is irreversible. You can't heal from long-term liver damage. Once that damage is made, then it's there to stay. And other conditions are correlated with heavy, frequent drinking, including high blood pressure, heart disease, stroke, cancer, digestive problems. The list goes on and on. But before I get into the details of what they've discovered in this research, I want to touch upon what constitutes binge drinking. How much does one have to consume for that drinking session to be considered a binge drinking session. And the National Institute of Alcohol Abuse and Alcoholism explains it's when a person's drinking habits cause their blood alcohol concentration, their BAC, to 0.08 grams % or above. Now let's put this in terms of how many drinks this takes, because that's kind of an arbitrary number. So for men, this is about 5 or more servings of alcohol. And for women, it's about 4 more servings. And that's all within a 2-hour period. So for men, they have to drink 5 in 2 hours. And for women, 4 in 2 hours. And obviously, that's just kind of a general statement. That's not too specific. But Dr. Shukla explained, quote, "Our last research shows that epigenetic modifications in histone structures occur within the liver as a result of heavy binge drinking. Epigenetic alterations are changes in genetics that are not caused by changes in the DNA sequence or genetic code." Now, that might be kind of confusing, so let's break that down. She's referring to histone structures, which are proteins that coil around DNA to help them compact into the cell. DNA is very, very long, and cells are very tiny. So in order for the DNA to really be packed in the cell, it needs these proteins to coil around. And the histone proteins also aid in DNA replication for when cells divide so that the body can grow or replace its cells and all of that, as well as expression of genes. And this research from the University of Missouri School of Medicine has revealed that unnatural modifications to histones result from binge drinking. So these histones that I'm talking about, these proteins that DNA is coiled around, are affected by binge drinking. So this includes DNA replication, gene expression, which means that protein production, as genes code for proteins, is affected. And that's a major part. So Dr. Shukla explains this concept further. Quote, "Every response in the body is due to alterations in proteins. Binge drinking is an environmental trigger that negatively affects histones by altering the correct binding of DNA." Again, that's like DNA to histones. "The result of unnecessary replication in the copied structure. This initially causes inflammation and damage to the cells as they form, but is also eventually the cause of more serious diseases such as cancer." And so she talks about how the liver is a primary organ to be affected, that is really the first organ to be affected for heavy drinkage. And this is not to say that other organs aren't affected, and they are. Dr. Shukla points out, quote, "It is important to specify that binge drinking should not be associated only with liver damage. Binge drinking can create an inflammatory response in the liver that is like a cluster bomb, sending out various damaging signals to other organ systems in the body. If those organs are working at a lower level of function, then a whole host of physiological processes are affected as a consequence of binge drinking." And binge drinking is a serious and prevalent health concern. The Center for Disease Control and Prevention reports that around 1 in every 6 adults binge drinks about 4 times each month. Let's just soak that in. Around 1 in every 6 adults binge drinks about 4 times each month. And again, binge drinking is when someone's blood alcohol concentration, their BAC, gets to 0.08 grams %, or we can think of it in terms of servings of drinks. For males, it's 5 or over, and for females, it's 4 or over within that 2-hour period. So hopefully with research like this, with more molecular evidence, that there'll be more attention on this issue. Obviously, that it is something that is a public issue that people are aware of, but hopefully research like this will amplify that, and people will see binge drinking as a serious health concern, because it is. And as I said, it's affecting 1 in 6 adults that they're binge drinking about 4 times each month. So this is something that's really affecting the public, and hopefully research like this will help highlight this, and then maybe even further we can figure out more of what's happening in more detail, of particularly these histones being affected by binge drinking, and understanding the effect of alcohol on the histones to a further degree. Now, this next news story isn't just about identifying one gene, kind of similar to before, that was about epigenetics, not really the bases. But this news story is talking about genes, not 1, but 400. In the largest genome-wide association study to date, an international research team has identified more than 400 genes that have an influence on a person's height. Now, this number doubles the previous research found, and doubles the previous number of genes associated with height found in research. And the data was acquired from more than 250,000 people. So this really is a large genome-wide study. And The conclusion they came to was that around 80% of height variation is genetic and the other 20% is influenced by environmental factors. So 80 of it is pure genetics and the rest environmental. And this 20% of environmental factor that we're associating height with, being environmental, is increased from previous studies that say environmental factor plays a smaller role. We're going from 12% previously to now we believe it's 20%. So this is increasing. And investigators shared that the height is a model characteristic for determining the mechanisms behind human genetics. Specifically, traits produced by multiple genes are understood better throughout this study. And the study is more basic when compared to other studies that explore multiple genetic traits, as height is simple to measure because you can just say, all right, this person is 5'11", this person is, 4'10", whatever they may be. This is much easier to look at, and obviously there's a lot of genes that put into play to see how this trait is affected, but the trait itself is very easy to measure, which makes this that portion of the research a little bit easier. And these researchers published their finding in the journal Nature Genetics, which is a good note to make. And 2 million genetic variants were studied that were present in at least 5% of participants. So 2 million of genetic variants were present in 5% of participants. Just wanted to really get that over to you. And after finding this, they identified that 697 genetic variants are located in 424 genetic regions that were linked to height. And a few of these genes that are linked to height were a surprise to researchers that they had not realized before. And one of those is mTOR, which is associated with cellular growth, but is now associated with skeletal growth as well. That before it was just, how our cells are growing and how much they're growing, but now we're seeing that it's not just cells, but skeletal growth. And Dr. Joe Hischler of Boston Children's Hospital and the Broad Institute of Massachusetts Institute of Technology and Harvard University in Cambridge, Massachusetts, commented on these new findings, saying that, quote, there were some pathways that we knew were important but had not come out in previous genome-wide association studies. Many of the genes were identified are likely to be important regulators of skeletal growth, but were not known to be involved until now. Some may also be responsible for unexplained syndromes of abnormal skeletal growth in children. As you increase the sample size, you get more biology. So there he's talking about how they studied so many people. I said 250,000 people that they took their genetic information from in order to really get a good sense of how many genes are affecting height. And obviously the more you're studying, the bigger your pool, the more accurate that research is going to be. That's all the time I have today for DNA Today. And you can tune in next week, Mondays at 1 p.m. and every Monday at 1 p.m. to learn more about genetics. You can follow the show on Twitter at DNA Podcast and shoot me email questions at info@dnapodcast.com if you have any. And join me next week, Monday at 1 p.m. and every Monday at 1 p.m. to learn and discover new advances in the world of genetics.